The COVID-19 Immunity Task Force and CanCOVID have partnered on a monthly event series, Research Results and Implications, to offer researchers, policymakers, and Canadians with the latest COVID-19 related results. To learn more about how both organizations have been helping to inform public health policy during the COVID-19 pandemic, please visit our websites at covid19immunitytaskforce.ca and cancovid.ca.
Welcome everyone to the 5th CITF CanCOVID Seminar Series event. This one in partnership with COVARNet. My name is Dr. Catherine Hankins, co-chair of the COVID-19 Immunity Task Force, and I will be your moderator today. Before we get started, I've been asked to share some instructions as we will have a bilingual question and answer period, and we are therefore providing simultaneous translation into English and into French. Bienvenue à tous et à toutes au 5e événement de la série des séminaires JTAC, CanCOVID, celui-ci en partenariat avec COVARNet. Mon nom est Dr. Catherine Hankins, co-présidente du groupe de travail sur l'immunité face à la COVID-19, et je serai votre modératrice aujourd'hui. Avant de commencer, on m'a demandé de partager quelques instructions car nous aurons une période de questions et réponses bilingues et nous assurons donc une traduction simultanée en anglais et en français. At the bottom of your Zoom screen, you will see an icon of a globe. Choose off if you are bilingual and want to only hear the live feed. English attendees, you should select the English option so you can hear the entire event in English. The panel discussion will be in English, but the question and answer period will be bilingual. When questions are asked in French, our panelists will answer in French, if a French-speaking panelist can answer that question. Au bas de votre écran Zoom, vous verrez l'icône d'un globe terrestre. Choisissez Off si vous êtes bilingue et vous souhaitez uniquement entendre le discours en direct. Participants francophones, sélectionnez l'option French afin de pouvoir écouter l'intégralité de l'événement en français. La table ronde serait en anglais, mais la période de questions et réponses sera bilingue. Lorsque des questions seront posées en français, nos panélistes répondront en français. Si un panéliste francophone peut répondre à la question. Veuillez sélectionner votre langue préférée maintenant. Please go ahead and select your language preference now. Shall we start with the next slide, please? And the next one. So we have, in addition to myself, we have Marc-André Langlois here, who is the Executive Director of COVARNet and Professor at the University of Ottawa. He's going to provide us with an introduction to COVARNet and to variants of concern. Next slide, please. We will also have 5 panelists who are going to be talking about variants of concern. They are all funded by the task force. Dr. Mark Brockman from Simon Fraser University, who is the Immunology and Vaccine Protection Pillar Deputy for COVARNet. Dr. Anne-Claude Gingras from the Lunenfeld Tannenbaum Research Institute at the University of Toronto. And she is the pillar lead for functional genomics and structure functions of variants of concern at COVARNet. Dr. Jun Liu is from the University of Toronto, and he's going to be talking to us about novel vaccines. Dr. Ciro Piccirillo is from the Research Institute of the McGill University Health Center, Immunology and Vaccine Protection Pillar co-lead, COVARNet. And finally, Dr. Jeff Mrna, who's the Lunenfeld Tannenbaum Research Institute, University of Toronto, Viral Genomics and Sequencing Pillar Deputy, COVARNet. So you see, we have a powerhouse here today to talk to us about variants of concern. Next slide, please. None of the panelists have conflicts of interest to declare related to the presentations today. Next slide, please. So just to give you a bit of an update on our mandate, the task force mandate, we were established in April 2020 by the Government of Canada. Our mandate is to catalyze, support, fund, and harmonize knowledge on SARS-CoV-2 immunity for federal, provincial, and territorial decision makers to inform their efforts to protect Canadians and minimize the impact of the COVID-19 pandemic. Next slide, please. These are our priority areas of research. We've had a big focus on seroprevalence studies that assess the extent of SARS-CoV-2 infection across Canada. Immune science, trying to understand the nature of immunity arising from infection. Immune testing to develop improved antibody testing methods. And vaccine surveillance to monitor the effectiveness and safety of vaccines. In our second year, we've gotten more involved with boosters, understanding and if and when different populations need booster shots. Pediatric vaccination, looking at the safety, effectiveness, and immunogenicity of vaccines for children under 21. And taking all the information that we have to put into modeling, immunity modeling, to model the trends in overall immunity across Canada. Next slide, please. So we're supporting studies across Canada, 112 studies at the moment, many of which include studies on variants of concern. Next slide, please. So it gives me pleasure now to turn over to Marc-André Langlois to tell us more about COVARNet. What is it and what is it doing? Marc-André.
Merci, Catherine, pour cette introduction. So here I will introduce all of you to what COVARNet is and what is it doing. Next slide, please. So the overall goal of COVARNet is really to unite Canadian researchers and researchers from every field, in government, in academia, basic, clinical, applied researchers, and bring them together and have them focus on the current pandemic and try to determine ways to help us out of this pandemic, and also to try to prevent what is another pandemic from happening and try to predict what is coming next. So more specifically, what is the network doing? It's attempting to rapidly and efficiently study the new and emerging variants. Most of these are variants of concern. So these are variants that can affect the health of Canadians. So our researchers are, their role is to characterize current and emerging VOCs and try to assess the risk they pose to the health of Canadians, both vaccinated, unvaccinated, and also our immunocompromised Canadians. The network members also are trying to predict evolutionary trajectories of the VOCs, try to figure out which VOC is overtaking the others and which ones will eventually will expose the population in the coming months. Also, our network members are trying to create new tools to analyze the variants, better ways to analyze them, also to share the data that is being produced, and also to create some shared infrastructure to facilitate rapid research. And then with all of this knowledge, with all of this information, our goal is also to share that information with the Canadian public, with researchers, so that we can all benefit from this research effort. Next slide, please. So COVARNet does not operate alone. It's a network that collaborates with other established networks in Canada, whether it be Genome Canada and its offshoots Kenkogen and CGen. It can be with the National Research Council, with CIHR, PHAC, CERN, also with the CITF, of course, but also with other Canadian and international researchers that may not be part of the network, but who can actually collaborate with us to fill in knowledge gaps. We also collaborate with industry partners. Next slide, please. So COVARNet currently counts approximately 50 members on its leadership committee and 90 members overall. And one of the major strengths of COVARNet is the multidisciplinary nature of the researchers that compose the network. If this pandemic has taught us something, is that we need various expertise to understand what's going on, what's coming next. And this group of individuals embodies this diversity and expertise. So the expertise of COVARNet is set up in different pillars. Next slide, please. And these pillars represent very focused areas of science, whether it's immunology and vaccine protection, host pathogen interactions, virology, functional genomics and sequencing. Next slide, please. We also have a team focusing on computational biology and modeling, how the variants are spreading in the population, a pillar for indigenous engagement, development, and research, a pillar that focuses on public health, health systems, and social policy impacts. This is a pillar that reaches out to decision makers and explains the research that is coming out of the network, a knowledge mobilization pillar that focuses on best evidence and its impact, and also a pillar on a new pillar on antimicrobial drugs and treatments. Next slide, please. So that gives you the overview of what COVARNet does and its expertise. Now let's get a little bit more into what are these variants, how do they emerge? So the first step of the generation of a new variant is that the variant that we see here on the left, this simplified virus structure, must enter the human body. It enters the human body through the respiratory tract, through airways. But this is not what we call an infection. An infection actually happens when that virus finds a target cell that expresses its receptor, in this case ACE2, binds the receptor, and the virus gets into the cell and then starts replicating. This is what we call the infection. And during the replication process, the viral RNA polymerase can sometimes make a few errors when it tries to copy that viral genome. We call these polymerase error prone, very unlike our own polymerases that are high fidelity. So by introducing these various mutations, the virus increases its genetic diversity, and sometimes some of these viruses are infectious and can then be released from the infected cell. Next slide, please. This is only the first step of the emergence of a new variant. Once the virus is released from those infected cells, it must tackle the immune system of the host. Now, the immune system can be the immune system of a host that is vaccinated, unvaccinated, or immunocompromised. And all of this landscape will help select the viruses that make it through. So this is how a variant emerges. Now, variants can be, but are not always, more transmissible, can evade immune responses, can be more virulent, or cause more severe disease. Some variants do not have these features. But if a new variant has these features, this is what we call a variant of concern. And these are the specific variants that COVARNet is trying to track. Next slide, please. Over the pandemic, we faced several variants of concern: Alpha, Beta, Delta, Gamma, and now Omicron. And what you see through this animation here is how they overcome each other. So variants that are more transmissible, are more successful at jumping from human to human, will eventually spread to the whole population. Initially, we had a variant that was the original variant coming out of Wuhan, and it slowly, and we saw the slow emergence of new variants of concern, initially Alpha, and then there was Delta for most part of last year, and now we are dealing with an Omicron variant of concern. But as you can see here, there's also the Omicron BA2. This is a sublineage of Omicron that is now taking over the original Omicron variant. So I will now return the discussion to Catherine.
Thank you, Marc-André, for describing the great work that COVARNet has been doing and for giving us some baseline knowledge about variants. We'll now move to the panel discussion, where I will ask the panelists some questions based on their research. Following the panel discussions, attendees will be able to ask questions of their own. You don't have to wait till then to enter your questions. As soon as you have them, put them in the chat function at the bottom of your Zoom screen. Now, my first question is going to go to Dr. Mrna and Dr. Langlois. This morning in Abuja, Nigeria, where the executive director of the task force is, the WHO Director General said that 83% of the population of Africa is yet to receive a single dose. So is there any link between that and the question I'm going to ask you, which is, where are the variants coming from? Dr. Mrna, do you want to start?
Yeah, thank you very much. I think that it's not clearly variants arise in regions where there's large levels of infection. That's correlated with alpha rising in Kent in the UK, and then the subsequent arrival of beta and gamma, et cetera. So the number of infections, as Marc-Andre described, the number of infections increases the chance that you will get variants that can do two things. One is to transmit much better, and the other is to evade immune responses. I don't think we really understand at this time exactly what it means for a variant to arise within a previously vaccinated or previously infected population versus variants arising in a naive population. So I think that's something where we really still need to understand what it is that makes a variant transmit.
Marc-Andre, do you want to add to that?
Certainly, a little bit. So the more we provide a variant an opportunity to replicate in different hosts, in different humans, and spread from human to human, we increase the probability of generating these viruses with mutations, new mutations, and new parts of its genome. We talk a lot about the spike, but there are 23 other proteins in that virus that each have a role in how virulent the variants are, how transmissible they are, how stable they are. So the more we let the variants spread and replicate, the higher the probability of generating new variants. And I think this is a major source of emergence of new variants.
So are you concerned about global vaccine inequity and its link to the potential generation of variants? I think it's a theme we'll maybe come back to during the meeting today. So my next question, and this is to Drs. Mrna, Gingras, and Picarillo, so in whatever order you would like. So the variants, they present us with numerous challenges, including not knowing how people's immune systems will react to the next one, how rapidly it will spread, whether the vaccines will combat it, whether our diagnostic tools will still work, whether our treatments are still good, etc. What would you say are the main challenges that variants are presenting? Who's most at risk, and can we stop variants? That's 3 questions. Let's see what we can do with those. Thanks. Who wants to start?
I think Jeff should go first, and then
Yeah,
Ciro, and then me.
Yeah.
Okay.
So I was just going to say, so in my lab, we systematically track most positives that arise within the Toronto area to try and understand the emergence of variants. And what's important, I think, for everybody to realize is the variants are arising all the time. So we've tracked dozens and dozens of variants that have arisen in the Toronto population. Many of them are completely novel. They target residues that we all know are immune evasion residues. These are residues that allow the virus to escape the immune response. But almost all of them actually in the Toronto area, they emerge, they circulate for a bit, and then they disappear. So I think that so variants are there all the time. So the answer to the first question is we won't be able to avoid variants. It's part of the biology of the virus. What makes one variant become dominant in the population, I think, again, is something that maybe I could hand over to the other panelists. The hard question.
Well, if you take a look at BA2 relative to its big brother, big sister, the traditional Omicron, it really has dozens of more mutations. And it's really that burden of mutations that determines the behavior of a virus. So what's the behavior of this virus? Well, it's really renowned for 2 things for which time will tell us whether these two things truly are of paramount importance. The first one is the increased transmission. This virus is remarkably transmissible between individuals, 1 to 2 times. I guess we could debate on the value of that. And the second is, does this BA2 variant really escape the capacity of an individual to become reinfected? Does it escape the immunity that one acquires through a prior infection, or whether it escapes immunity acquired through prior vaccination? There is emerging evidence to indicate that BA2 may have a slight advantage in that escape potential compared to traditional Omicron in terms of the capacity of the circulating antibodies to stick, bind, and neutralize that virus. But I think there are also studies that indicate that that may not also be the case. So the immune escape potential is of extreme importance. I would even add that this mutation is of particular importance because it can actually escape the protective effect of some of the available, what we refer to as biologics, antibodies, monoclonal antibodies that typically are given to individuals to limit transmission and disease progression. So immune evasion is always a major concern for any variant of concern that emerges in a given community.
So maybe that was kind of what the third question was supposed to be, more about BA2, but I mean, we can use it as a good example because Omicron itself got picked up early in South Africa because of a diagnostic thing, I think, this S gene target failure on the regular PCR testing. And maybe Anne-Claude can explain that. And then this BA2 doesn't have that. So it's evolved to be, not having that. So we can't use that for early detection. We have to do the sequencing and so on. So, Anne-Claude, where are we at with, just use this as an example, the BA2. Is it going to become predominant in Canada? Is it getting there? And should we be concerned? Should we be worried about it? We know that Omicron, the first variant anyway of Omicron, was evading immunity, and we only had one monoclonal left in the ones that we were using for treatment. So what can you tell us about where we're going with BA2?
Right, so just to focus on BA2 versus BA1, first of all, both of them are still referred by the WHO as Omicron, right? So they're lumped together in this big Omicron category. So when we talk about Omicron, it's both BA1, BA2. So first, I just want to comment that the WHO still thinks that they're similar enough in their properties to jointly refer to them as Omicron. And the properties that they're looking at in this case is both the transmissibility, which was very much increased in comparison to previous variants. Now, it is true that BA2 seems to have a growth advantage and maybe a shorter incubation period before being contagious compared to BA1, but still, if you look at both of them together, they're still much more transmissible than previous variants, including Delta, that was itself very transmissible compared to previous variants. So in terms of immune escape, again, there's a different mutation pattern between Omicron BA1 versus BA2. They're in fact as different from one another than the Alpha variant was from the wild type in terms of mutational load, right? This being said, if you look at the serum, the blood of vaccinated individuals or individuals that have recovered from previous infection, their ability to neutralize, to prevent viral entry of either BA1 or BA2 is pretty similar. Chiro mentioned maybe a little bit worse for BA2, but this is very, very small numbers here. So again, like in terms of their ability to neutralize, to evade the immune system, they're very similar. So again, the WHO thinks like these things are similar enough to be bunched together. The third point that we haven't really discussed is disease severity. And this is a very, very important point because you want to know whether you have evidence that people that have infection with variant A versus variant B, in this case BA1 versus BA2, have a different outcome clinically. So now this is a bit early still for BA2 versus BA1, But right now, some model experiments using animals, right, they're not humans, indicate that BA2 could be a little bit worse. But the results coming out of Denmark right now, which has the highest proportion of BA2 cases after a big wave of BA1, have not reported any significant differences in clinical outcome. This is true also for South Africa, that has a different makeup of vaccinated and infected people. And this seems to be also true in the UK, that has a very active surveillance system. So again, if you just compare BA1 versus BA2 right now, they look quite similar in many properties. There are differences, and you made the point of the monoclonal antibody therapy. That's you're referring to a particular therapy called sotrovimab, which was able to prevent viral entry of BA1 and is much affected, much reduced in its ability to prevent BA2. So again, like this is of concern for therapeutic treatment of people that end up being in hospital. But there is a point I want to make, right? The best way to prevent yourself from being sick from BA1 or BA2 is to really get your vaccination on schedule. And if you're eligible for a third dose, this really boosts neutralizing antibody response to both BA1 and BA2. And this is true not just in the studies that Covarnet has done, and both Mark Brockman and myself have done several of those studies, but this is true in studies across the world. This is true in real life scenarios, right? People that have been triple vaccinated, have 3 doses of vaccine, are much less likely to get infected still by Omicrons than people with just 2 doses of vaccine. And if they do, they're much more likely to have a milder form of the disease. So I think that's important to keep in mind, even though we say in the news like, oh, neutralization doesn't work for Omicron. You know, it is much reduced, but the booster dose really helps. And I'm going to stop here for this.
So that's actually a great segue into our next discussion, which is about vaccines. But before we go there, can someone please comment on the differences between Delta and Omicron in terms of pathophysiology, that Omicron tends to focus more on the upper respiratory tract? Does anybody want to comment on that?
Marc-André should take that one personally.
Well, this is one of the main observations with Omicron, and why partly explains why it's so transmissible, is that it focuses on the upper respiratory tract, and the vaccines and also infection-acquired immunity, the antibodies we develop, are mostly protective in the lower respiratory tract. So this virus can actually enter the body quite easily and establish the first infection in the upper respiratory tract. And this is a feature of Omicron that we are seeing that is slightly different to that of Delta.
In a way, thank heavens. Okay, let's move along and look at vaccines. They're doing an excellent job, the current vaccines that we have, in protecting everyone from hospitalization and death. I mean, the general public, not people that are really highly vulnerable, but the general public. They've done an impressive job protecting us from previous variants. They're doing a modest job of protecting people from infection from Omicron. In our last seminar, we discussed waning immunity and the need for boosters. But as we look to a future with potentially more mutations and variants, do we need to look at different vaccines? Now, Dr. Liu, Dr. Langlois, Dr. Brockman, you were among the Canadian researchers trying to develop a multivalent vaccine, a vaccine that's able to combat any variant. In fact, I think WHO was talking about the importance of a pan-sarbecovirus vaccine to handle all of the coronaviruses, because there are so many that are resident in bats, the beta coronaviruses. And as we've seen, there are various zoonoses where it jumps from an animal or whatever to humans, and then from the reverse zoonoses going back. So what we want to talk about today is now the idea of a vaccine that would be sprayed in the nose instead of a shot in the arm. So can you talk more about why these vaccines are important, how far we are from using them, and why a nasal spray could be more effective than an injection. So I think, Dr. Liu, we'll put your slide up first and talk about that, and then we'll expand to Dr. Brockman and Dr. Langlois.
Yes, thank you. Can I have my slides, please? Yes, so thank you, Catherine, for the introduction and the question. You're absolutely correct that the currently used vaccine have done an excellent job protecting us from developing disease and becoming ill. They are less effective preventing us from infection. And this is partially due to the fact that all the currently approved vaccines are delivered by intramuscular injection into the arms. While this induces very strong systemic immunity, including, for instance, antibody and T cell in your blood, which results excellent protection of the lower restricted tract, for instance, our lungs, our hearts. But this route of the vaccine delivery in general do not induce a local immune response in the upper airway, such as our nose and the mouth, which is where we normally get infected. And this is perhaps why we have, as my colleague just mentioned, we have more infection of the Omicron in the upper airways. In contrast, next slide, please. A nasal delivery of the vaccine can generate both systemic and local immunity. For instance, the secretory IgA in the upper airway. And as a result, the vaccine developed by either a nasal spray or intranasal inhalation will have all the benefits of intramuscular injection, but will have additional advantage of being able to prevent infection in the upper respiratory tract, which would, in theory, prevent the virus spreading and the transmission. So I know that some companies are currently developing the nasal spray technique, and I think we were likely to see them in use in the near future. Next slide, please. So the project we are currently working on is trying to develop an intranasal multivalent vaccine based on the helper-dependent adenoviral vector. One of the advantages of this HDAD platform is that we removed all the adenoviral gene in the vectors, which allows us to clone multiple transgenes in one vector. So for example, in this case, the S genes from 4 different variants, including the Omicron variants. This would allow us to target multiple variants in one vector. The HDAD platform also has a long-lasting capacity to produce antibody and T cell response, which has a potential to provide long-term protections. As for the universal vaccine for all future variants, some of the ideas have been proposed in the research field. For instance, one could design such vaccine based on conserved epitopes of the S antigen in combination with some other antigens that undergoes less frequent mutation. So far, our vaccine, most of our vaccine have been focused on S protein, but there are many other antigens that can be candidate for the vaccine designs, as well as another idea would be develop a vaccine that targeted T cell response, for instance, CD8 T cell response rather than the antibody. But of course, much research is still needed in this area. So I like to hand them to my other panelists.
So, Dr. Brockman, don't we have a nasal vaccine for influenza? I mean, is that part the idea of presenting the antigen nasally or mucosally? Is that novel, or have we got some experience with that kind of thing?
No, you're correct. I think we do have good experience with generating nasal vaccines, and some of them that have gone through full Health Canada approval and are available in the population. So, I think Dr. Liu's work is excellent, and really, I think are the first steps to getting us into a type of platform that could be more easily delivered. But definitely, the technology is available.
It's exciting. It's really exciting. So I just wondered, Dr. Liu, do you want to comment more, or should we ask? I think we were going to ask Dr. Langlois if he wanted to comment on this, whether, and the idea of, both neutralizing antibody and T-cell immunity in one vaccine. That sounds exciting. Marc-André?
I think it's also important to recognize that these are not alternative approaches to vaccination. They're complementary. So the current vaccines are outstanding to prevent severe disease. And I think those vaccines should continue to be deployed. These intranasal vaccines offer something additional to those vaccines, is that they are highly neutralizing at the point of entry. So here we are dealing with a respiratory virus. It enters your airways, and that's where you want to have a neutralizing, a sterilizing immune response, so that when the virus enters your nose, enters your mouth, you will have antibodies there, and you'll have immune cells there that will prevent the virus from even entering the first cell and causing the initial infection. So I think this is where these intranasal vaccine technologies really excel, is to focus on preventing transmission.
So you could maybe see it as you would have both. You might have your shots and then maybe a nasal boost or something, depending. And what's interesting about what you've presented there, Dr. Liu, is the possibility of modifying according to the variants, the epitopes as needed according to what we're seeing. So that sounds like it's more flexible perhaps than what we're currently using.
That's right. That's basically the idea we are trying to develop, and based on this platform, because we remove all the adenoviral genes, so we have a large space, we can clone multiple transgenes, which allows us to potentially introduce the representative antigen from different variants in a one vaccine. We have a potential to protect multiple variants. And I should also say that I agree with my colleague about the advantage of the retronasal or nasal spray is the protection at the site of infection. This is not, we're not trying to replace the current vaccine we have, but it's just basically has additional advantage. Some of the major benefit is, for instance, the secretory IgA that's been produced in the upper airway that really helps to prevent the infection at the site of the infections. So.
Okay, great. Anybody else want to chip in before we move to the Q&A? Because we've got at least 18 questions that have been put in the box.
I can add just to the conversation that was also presented by Dr. Liu, is that a lot of our focus in the community and as researchers has been on the antibody responses to the virus, whereas if we're talking about a pan-Sarbeco strategy, we really want to be focusing on the highly conserved parts of the virus, which might be conserved aspects of the spike protein that can be recognized across these variants and strains, but also moving more into the T-cell responses that are much more conserved from virus strain to virus strain. Will be an important aspect of these new vaccine strategies.
Mark, while I have you, you were going to get the last question before the Q&As, and that was, where are we? I mean, we all want to know, where are we in this pandemic? The question was, what is endemicity? What would it look like? What is a world with constant variants of concern? What does that look like? Can you give us something from your crystal ball?
Yeah, I'd say it's impossible to predict, but I think that it alludes back to the question I was asked, I think, at the very beginning, is as long as there's inequity in vaccine delivery around the globe and people continue to be infected, there will be a continued risk of generating new variants. And so I think that endemic, whatever that means in Canada, will be a lot of it will be dependent on what's happening globally. We can't keep the virus or new variants of the virus from coming to Canada or spreading around the world. So I think we need to be expecting that new variants will occur, and we will see them showing up in Canada. So I think, again, the best thing we can do now is to get as many people vaccinated and boosted as we can. Our own data from our own lab, and Dr. Gingot alluded to some of this earlier, is really suggesting that a third dose of vaccine is quite effective. So I can remind people that when we started this whole process, we were looking for a vaccine that provided 50% protection from infection and disease. And we still, with the current vaccines, we're seeing much better than 50% protection from infection, even against the Omicron strains, and even 80+% protection from severe disease. So I don't think we're in a bad place. I think the vaccines are doing their job, but we do need to be prepared for what comes next.
So basically, and this endemicity doesn't come for us until it comes for the whole world, basically, to keep that in mind.
No, I think it's an important point.
Let's go to our questions. So to whoever wants to ask this, we have the first question is, what would be the biggest game changer for reducing the time from when a variant emerges to having good epidemiological information on its impacts? And we don't need to have a slide on that says questions. It's okay, you can take that away. Then yeah. I can see actually who wants to answer. Jeff,
I can answer that. Yeah, so I think the way you can track that is to is by basically surveilling the population for the emergence of these variants within the population, also tracking how they spread within the population. And so, for instance, as I mentioned before, we see variants emerging all the time in the population, and so we can really track them in a rapid way, almost on a daily basis, to see how they expand and then how they spread within say an urban population. So if a virus might arise, for instance, in a particular forward sorting address, the first 3 digits of your postal code, but never expands beyond that FSA. So it's probably more like a kind of an outbreak kind of thing. And by tracking that on an intense basis, then you can actually see whether the virus is going up and keeping going up, or whether it's going to plateau and go down. And like I said before, for all the variants that we've seen emerge in say the Toronto population, they've all expanded and then went extinct. So we track that all the time.
Okay, so there's 2 questions. I'm going to ask Anne-Claude maybe to answer this, but one just relates to something Jeff has said. So the person has written, I've heard that there's a finite number of possible mutations. Does this mean that eventually COVID will disappear because the whole gamut has been spanned and we've built up resistance? So Anne-Claude, what do you think about that? This is another question for you as well about variants. Are variants only on the spike protein? Or do you look at other aspects of the virus? And are variants always mere errors, or are there evolutionary pressures to evolve? So those 2 questions kind of come together.
Sure. So let me start, and I'll pass to Marc-André, because I think for the first bit of the question, he's in much better position than me based on his track training. So the question is, are there variants only on the spike protein? The answer is no. So people have put much more effort on understanding the variants on spike, because spike is responsible for viral entry. So blocking viral entry has clear and obvious implication in essentially blocking infection, eventually also blocking severe disease. But there are variants in other proteins. So there's 20 additional proteins in spike. Some of them in SARS-CoV-2, some of them are frequently mutated in variants of concerns, and the mutation gives the virus some additional advantage. And the advantage can be, again, to contribute a little bit to immune evasion in some cases, but often some of the advantage will contribute to a more efficient replication or a worse pathogenicity of the variants. So in CoverNet, we're tracking those variants as well, and we're doing different type of experiments to go after the different proteins. For example, the nucleocapsid, which is one of the structural proteins of SARS-CoV-2, has a very characteristic mutation that we see in almost every VOC that has emerged, and it's still poorly understood exactly how they contribute to the worst pathogenicity seen with those mutations. Same thing with some of the non-structural proteins. So we do look at them. and we look at them also in the context of the entire virus, right? So if you isolate a virus strain, BA1, BA2, you have Delta. If you do experiments that are going to use this entire viral strain, then at the same time, you answer a question about every mutation that you have within this variant strain. So I think I want to leave it at there for the other variants. I think they exist. And then Marc-André can cover the evolution of viruses a little bit better.
Yeah, so Marc-André, if we can get back to this question, I mean, will COVID simply disappear when all of the gamut of mutations has been spanned? You know, when we've built up resistance, and what do we mean by viral fitness? People talk about the virus being fit. What does that mean?
Well, it's a broad term. So I'll just back up a little bit. So the virus cannot mutate indefinitely. So mutations have consequences. So a mutation can help the virus in some manner, help it replicate maybe a little bit faster, evade immune restriction. But mutations can also be disfavorable for the virus. Some mutations can actually inactivate the virus. So those viruses end up never being released. They just do not replicate. And some mutations are neutral. We do see these types of mutations, but they tend, if they're not useful for the virus, they don't tend to stick around for very long. When we talk about fitness, these are mutations that enable the virus to persist and spread. Doesn't necessarily make the virus more virulent, so cause more severe symptoms. It allows the virus to basically be able to spread and in the local demographic where it's located. So a virus, for example, that is spreading in a immunological naive population will have certain features. But a virus that is spreading in an immunized population with 2 vaccines or 3 vaccines will have various other features. It might sacrifice its ability to replicate to be able to counteract neutralization. So it's always a balancing act for the virus to persist and continue to propagate, and also to ensure its own replicative ability. So it's always a balancing act for this virus. Will it disappear? Well, given how widely spread it is across the globe, and given that a very, very large percentage of the human population is unvaccinated, we can expect this virus to be around for a very, very long time. Will it ever disappear? I cannot answer that, but I can guarantee we will be dealing with this virus for a very, very long time.
Okay, thank you. Now, a very technical question, and I don't know who wants to answer. If stringing multiple transgenes in one vector, how will you ensure proper protein folding? I have no idea the meaning of the question, but perhaps somebody can answer that.
And Claude,
Yeah, so,
or go ahead, June.
June, do you want to die?
June.
Yeah, so that's certainly one of the aspects when we design the vaccines. I won't bore you with all the technical detail, but it does require certain expertise. So we have a structural biologist, Dr. James Rini in our team, who is helping us for that kind of design. But it will take much longer time to explain this, but I won't go.
Maybe with the person who's asked the question can get a hold of you and get more details. So we do have a number of questions about vaccines, and there's a key question I think that's important for everybody to understand. It's about the effectiveness of mRNA vaccines. So it's saying basically the traditional vaccination effect can completely protect from diseases such as polio and measles and so on. So why are these mRNA vaccines only allowing disease expression to be diminished? You know, there's some effect on infection, yes, but we're really looking at preventing severe outcomes and deaths and so on. And so what can we tell us a little bit? I don't like to use the term, which we use all the time, called sterilizing immunity, because I don't want people to get confused and think it has anything to do with fertility. But maybe somebody can explain for the person who's asked this question, what's different about what we're trying to do with these mRNA vaccines, and why can't we get elimination, eradication, or something with the vaccines we have? who wants to take off their mic and take that? Okay, Marc-André, what do you?
I can start this. I think it's not a question about the vaccine. It's a question about the pathogen. So there are, the blood transmitted pathogens. I'm an HIV researcher. We can think of a virus that is sexually transmitted, one that's inhaled, one that's ingested. So there are various ways by which a pathogen will enter a host. In this case, we're dealing with a respiratory virus. So it comes in through the nose, the airway, the mouth. And if we want to prevent transmission, that's where the initial block needs to happen to prevent that transmission. So the current vaccines, what they're doing is that they're stimulating our immune system, they're educating it to recognize the virus. But once the virus enters, the bloodstream or the lower airways where the cells, the immune cells are located, once they see the virus, they're going to act on it. But the type of vaccines that we have right now do not prevent that transmission. So an mRNA vaccine might be extremely efficient in preventing transmission and disease with other pathogens. And we're hearing about mRNA vaccines now being designed for HIV. And there is a possibility that that type of vaccine will prevent transmission.
Okay, so it's not the platform. It's actually the virus itself.
Correct.
And it's a virus, with, unlike smallpox, there's asymptomatic carriage of it, asymptomatic transmission. It has many challenges for us. So question for you, Junju, would nasal vaccine have less adverse effects than something that was intramuscular? Are there disadvantages to using it? And the question also was a bit answered already by Marc-André about could you use intramuscular and then nasal and so on and so forth. And then that links up with another question that's about intranasal influenza vaccines not performing well. So maybe let's start off with, first of all, adverse events from nasal vaccines.
I think the whole question depends on the design of the vaccine. So one cannot generalize saying intranasal is safer versus intramuscular or the vice versa. It's really depends on the specific design of the vaccine, the formulation of the vaccine when it comes to the adverse reaction. So based on, at least in our cases, our study in the animal model, the intranasal delivery did not have any adverse reaction compared to the intramuscular. There's no disadvantage compared to the intramuscular, and potentially safer actually than the intramuscular injection using the platform we are using. But again, I should emphasize this is specific to individual vaccine formulation. One cannot generalize a conclusion based on just like a general statement. As to, again, why the intranasal vaccine for the flu did not work as well as a conventional vaccine, again, I think it's also depends on the disease. I sort of agree with my colleagues mentioned earlier, one cannot compare the vaccine effectiveness between different disease. It's really depending on the pathogenesis of the virus, of the bacteria, or the pathogen, the mechanism, how they cause the disease. In fact, the host cell are totally different. So it will really depend on how the vaccine was designed and how much we know about how this pathogen will infect and cause a disease. And it's rather specific. We cannot generalize, make this kind of a general statement.
Okay, thank you. I just realized what the time is, and we do have a number of French questions that we want to see answered. So, nous avons plusieurs questions en rapport avec la surveillance des eaux usées dans ce temps de variants. Marc-André, quelle est l'utilité du monitoring des eaux usées pour suivre l'évolution des variants? A-t-on besoin davantage de stations de mesure? J'ai aucune idée combien on en a. Est-ce qu'un programme national serait souhaitable? Marc-André.
Est-ce qu'un programme national serait souhaitable?
National program on this huge water, yes. what happens is during this pandemic, we discovered that the monitoring of this virus in the sewage water, that is a genome of the virus, permits us to predict the trajectory and the infection of which is certain variants go which way. That is usually 5-6 days delay between the what we see in the sewage water and what we see in the hospitals. It permits us to install the monitoring system to see what is going to happen, and also the monitoring of sewage water also permits us to see it on the level of the total population. That's very good, because we cannot get the sample from everybody, but well, this is the way to see the population in general, and also to see the emergence of new variants which we don't know about yet. We do the sequencing and the monitoring, and it permits us to see the new emerging mutations and those which could eventually constitute a new variant and would affect the population. So I would say, well, the sewage water monitoring is a very good desirable thing. Next question. If the number of persons infected by the virus in the society goes up, gets to the plateau and then goes down, is it the virus itself, or is it because of the vaccination and the measures? That is, without the vaccination and measures undertaken, would it do the same? Well, it's a little bit a charged question. Yeah, very charged as a question. There are so many variants and variables that influence the transmission of the community transmission and the levels of the efficacy on the expression of the disease itself and the contraction of the disease with the time. It's very clear that sanitary measures do play a very important role in order to minimize and to decrease the transmission, the community transmission. And evidently, the vaccination level is also one of the main factors. The vaccination and also the boosters, they are really able to minimize the capacity of the virus to replicate itself in the individual and to get transposed from one individual to another one, from one person to another one, and to develop the disease. There are also other factors that are neither sanitary measures nor immunitary measures, but which are not known maybe, and which are maybe known, but very little explained. For example, the seasonability of the virus. That is why, for example, the influenza virus is very predominant in the spring and in the autumn, and why it's less expanded, for example, in July. The same thing is valid for the big family of the viruses, coronaviruses. And for example, look at the April and May. This wave is also happening naturally and declining naturally in April, May, and doesn't depend. That part doesn't depend on the vaccination and the sanitary measures. But the level of vaccination is also one of the most important aspects right now during the pandemic time. Once again, another reason to go and get the third dose if you didn't do it yet. I think that we're going to finish soon. We have only 2 minutes remaining.
I unfortunately have to use the remaining time to make a few very brief announcements. I want to say that we will try to answer all your questions. There were so many great questions that we just couldn't get to in this short amount of time. And our immunity task force will work with the panelists to answer your questions. So you should be getting information about that shortly. So on behalf of the task force and CanCOVID, I'd like to thank today's speakers and attendees for sharing your time with us and for contributing to this important discussion. We know how busy you all are, so we appreciate you taking time out of your day to join us. If you can put up the slide promoting the document, our next event in this series will be a pediatric vaccination. It should be a very interesting event, so please stay tuned. Sorry, the slide before was of our panelists for today, just to remind you of who was speaking today, great panelists. And the next slide there will be a summary document that will come out of this discussion today. So stay tuned. And if you're interested in any of the publications referenced in today's event, you can find them listed on the task force website and at the end of this event's recording. Thank you again for joining us, and stay safe, everybody.
We would like to thank today's speakers and attendees for their time, as well as the members of our networks for their continued support and participation in Canada's pandemic response. If you are interested in attending our monthly research results and implications events, please visit either website for more information.